share with you the difficulties we have and the things we learn, so you can move faster than we moved. But that's about it. So if at some point you know something that we don't know, please jump in, and we will appreciate any input that you can provide. So Tyson will start going through the presentation, and I will make comments in the middle.
Okay, so like George said, we're going to be talking about what we learned with the canopy spectral reflectance using our Jaz unit. We just kind of been doing anything we can think of. Like you said, we're not experts. Here's a picture of our equipment setup. This is George and one of our students that we have working here. You can see just have the Jaz and the Jaz tag, so you can hold it around your neck. It makes it a little bit more comfortable. And see if I can get the pointer to work here. Maybe not. Oh, there we go.
Yes.
So this is our pole, which is just made out of 3/4 inch steel pipe right now. It's just something really quick and simple we got from the hardware store. It seems to work pretty well. Sorry, I'm working on a small screen here. This is a close-up of just the end of it where we have the two fiber optic detectors. We have our downwelling channel, which is pointing up, so it's looking at the sky, kind of recording the incoming light for us. This little black thing on it is our light diffuser, so that it's not getting light just from one tiny spot in the sky. Down here is the upwelling. So this is what's actually recording the reflectance from our plants. And as you can see, we just kind of have these held on here with string and tape. It's a little bit crude, but it works for us, and it actually did a pretty good job for what we needed to do.
Yeah, notice that can you go one before, Tyson? Notice that we ordered the fiber optic cables with this metallic cover. It's a little bit more expensive, but I think it's worthwhile because it's really well protected that way, and it's very flexible to have.
Yeah, it's definitely something you should get, just because you're going to be carrying this thing all over the place, and it's really easy to break the regular laboratory grade fiber optics.
And the other thing is that for the CCC3, can you go back 1 second? This one doesn't come with a lid, so we just use a pencil cap, and it fits perfectly and protects this diffuser. How it's called, CC3 or something like that?
Yeah, I think it's a CC3 detector or diffuser, something like that. Okay, next slide now. Okay, so this is a picture of the white reference that we've been using. So this is what we use to set reflectance at 100%. Some notes on the bottom of this. They seem to be gone now. We just bought this from Office Max. It's just a regular foam core board. I think it's Elmer's brand. It's 20 inches by 30 inches, so it's pretty big. But it is kind of nice to have it a little bit bigger so you can play around with it. Sometimes the sun will get into a position where there's shadows in certain angles and stuff, so it's nice to be able to move it around a little bit. And you can see we have a little bubble level down here. This is just so we can make sure that it's completely level before we take our reference. Oh, and one more thing about this. We do plan on making a barium sulfate white reference, but just as a quick kind of fix, we use this and it worked out all right. I think it'll be better to do the barium sulfate to compare measurements between the different groups, just because if you buy different kinds of foam core, there might be slightly different colors and that kind of thing, which might throw off comparison between different places. Here's a quick output from Jaz. I highlighted some of the key things here. So at the top of each file is a date and timestamp, which is useful to have. It just kind of helps you keep track of things. You can see the order that you took the readings in. And then we have some other stuff in here. There's the spectrometer name. You might want to keep track of that if you're going to be using the two channels, the upwelling and the downwelling. It just tells you if you took a dark, if you took a reference, if you asked for the process spectrum down here. It's all just kind of default stuff. The integration time is basically the exposure time. It's the amount of time that the detectors were collecting information from your sample. So the higher the exposure, or the higher the integration time, the more quality data you're going to get because it's averaged over a longer time. But at the same time, it's really easy to saturate it.
Let me say a couple of. I want to say, a couple of things about the integration time. The way at least our Jaz equipment was configured by them is that when you click the bottom, you get at the same time the upwelling and the downwelling measurements. So you get two files when that click of the bottom. But the problem we are having is that for now we cannot change independently the integration time. That means if we select an optimum type of integration from the light that is reflected, usually we saturate the other one. So what we end up doing is we take specific measurements for the upwelling with an integration time, and then we take different measurements for the downwelling with a different integration time.
Yeah, and you'll kind of see that on this next graph I have down here. But yeah, just the rest of the things on here real quick. There's something for the averages. We usually set it to 10, so that means it takes 10 readings all at once, and then it averages those together, and that's what you're actually seeing for the rest of this down here. So this is averaged from 10 different readings in the same spot, and it's really quick. Here the integration time was 35 milliseconds, so it takes 35 times 10 milliseconds. So it takes a third of a second to take 10 readings all at once.
We played a little bit with the number of applications before, but 10 is sufficient. So we settle on average 10. So that we use now 10, average of 10 for everything we do.
Yeah, and then we take, we've actually been taking 5 different measurements over the plot. So we're really averaging together 50 measurements when we actually do our analysis. Down here we just have correct for electrical dark and correct for detector nonlinearity. Those are basically just correcting for differences in the way the spectrometer behaves due to different temperatures and that kind of thing. They talked about that a little bit at the CSR forum in Colorado.
I think that you skipped the boxcar smoothing.
Oh, yes, I did, sorry. So boxcar smoothing, this basically just smooths over your spectrum data that you're getting. A boxcar of 1 means that it takes your a given wavelength and it looks at one wavelength on either side of it and it averages together those 3 wavelengths. So if you have a boxcar of 2, you're actually averaging together your 1 wavelength plus 2 on either side, so that's a total of 5. We decided to set it at 1, so we have a little bit of smoothing in our raw data, but for our actual process data later when we're looking at the indices, we've been doing closer to 10. And we played around with a little bit. It doesn't seem to make big differences, but we'd like to keep our raw data as true to what we actually measured as possible. So that's why we have it set at 1.
I would like to go a little bit deeper in this one because we spent several days on this boxcar smoothing. So first of all, if you look at the column on the left, this is the wavelengths that we are collecting. So in our case, of our spectrophotometer is set up to collect between 500 and 1100 or 1200. So what you're seeing in this first column that I'm marking with the green arrow are the wavelengths. And as you can see, the spectrophotometer is taking 2,000 measurements. So it's taking more than 1 measurement for wavelengths. It's actually taking 1 every 1/2 or 1/3 of a nanometer. So when you actually make a smoothing of a boxcar of 1, look at the green arrow right now that is in 493. Actually, what it means that I will, using a boxcar smoothing of 1, is that I will make the average of 493.2, 492.9, and 493.6. So I will make the average of these 3 measurements, and this is the number that I will get here. So this is an average of a window of measurements. What happened is at the end, when we decide the measurement that we use in the indexes, we also take another window. So for example, if the instrument says, well, take the 900 nanometer and divide it by the 970 or subtract the 970. In that case, what we do is we again will take a window around that. So we are already making an average later. So that's why there's no point to make a big average at the beginning, because if we want that average, we can make it later by taking a larger window when we make the average. The problem is if you start making your boxcar a little bit too big, if there are peaks that are too tiny, that they occur in a very narrow wavelength, the smoothing will start averaging those and will start reducing. So you can see my hands now, so you can understand what I'm saying. Any questions for that?
George, did you see Mary has a question there in the chat box? She wants to know how you're measuring dark so that no light comes through. Cap on, finger over.
Yeah, we've been doing that. We just put the cap back on. If you saw in the picture earlier, there's a little blue cap tied up here. We just put that guy over it and take the measurement that way. Okay, so back to the output slide. Just a couple other quick things. Here, this column is the measurement where we took the dark reading. So this is what we used to correct for just kind of the electrical noise. This column is for the reference. That's the measurements that we got off of our white reference. This is the sample column. So this is all the values that we got when we took our actual reading over the plants. So these first 2 columns are going to be the same for a given reference that you took. And over here, the P column is the process data. So this is actually in terms of percent reflectance. And these values look kind of weird because they're at the top. These are actually below the threshold of what our spectrometer was meant to detect. We have a filter on it that blocks anything below 500 nanometers. So this is like what you're seeing here is basically all garbage data. But as George was saying, there's 2,048 data points in here. So this is just the very beginning. It's really noisy, and we don't use it anyway.
Let me mention a couple of things about the formula that you're using, so you understand what that p-value is, because that's the one that we are going to be using in all the analysis. So that index is S, that is the measurement that you are taking of the energy that is reflected at that wavelength, minus the dark, so you are subtracting the noise, and then you are dividing that by the white minus the dark. So basically what you are saying is the reflected light as a percentage of the maximum reflectance that you can get in the white board. So this is the process data. It's a percent reflectance value. Questions for that? Okay.
Okay. Looks like Mary's typing something. We can answer that when she's done. Here's
Mary
yes.
here has a question. Is the process data
that's
what
correct.
shows up as the spectrum on the display? Yes, it is.
Yeah.
Exactly.
So right here.
Exactly as it shows.
Yeah. This is.
Sorry.
Okay.
This is just a file, a normal file. It's a flat file, and this is a tab separated column. So you.
Okay, looks like we're just going to be busy with this phone call now. So down here we have an example of our collected data. So this is the process. Yes, the plot is P. It's in terms of percent reflected. So if you remember from our CSR training in Colorado, this is basically what the display looks like on the Jaz when you're taking measurements. And this is basically the P column graphed over wavelength. So this scale here is in terms of percent reflectance. So it goes from 0 to 100. And down here, this is over different wavelengths. So this is each individual wavelength graphed with its percent reflectance. And this is basically how we keep track of all of our plots. So as you can see, we have a dark that we took here. So this is how we know we ended one plot and we're going to begin another one. And then we took 5 different measurements from our next plot. And then we blanket again with the dark so that we know that we've gone from one plot to the next. And it's really important to do this because it's really easy to just kind of push that button twice and take 6 measurements instead of 5. And if you do that, then you start averaging measurements from plot 4 into plot 5, and it just kind of messes everything up. So it doesn't take that long to do, and it really helps later with the data analysis.
Let me say something. So here what we are doing is we go through the plot, and we walk one step, take a measurement, walk one step, take a measurement. So basically, we leave, I don't know, 50 centimeters at each end of the plot, so we don't touch the end of the plot, but then we cover all the central part of our small plot with 5 overlapping discs. Our height was around half a meter over the surface of, at the time we take these measurements, there were some spikes also, so it was, to the bottom of the spike was around half a meter. That means that is our circles of 22 centimeters. So basically we are taking 5 circles of 22 centimeters diameter, and you can see that the plot is very consistent all across the plot. So you get very, very similar results.
Yeah, and one other thing is sometimes we had areas where the plants were a lot less dense. You could kind of see down to the soil, so we tried avoiding those as much as possible when we were doing this, just because they mess with the index a little bit. You'd think we haven't done extensive testing, but yeah, we were watching out for that while we were doing this. Okay, so here's a graph of our downwelling values. So it's a little confusing when you first look at it, but basically this red line up here is the intensity that we were measuring at wavelength 880, which is one of the wavelengths that we use in our water indices. This blue line is wavelength 970, and this green one is 900. So these are all 3 different wavelengths we use in our indices, and they're graphed over the day. And this is just the downwelling. So this is the incoming light, and this is basically, we're just looking at how the light is changing over the day. So you can see it starts a little low in the morning, and then around like noon to 1, it gets brighter, and then later on in the day, it starts tapering off, which is kind of what you would expect, since it gets not as bright in the morning, gets brighter around noon, and then starts getting darker later in the day. So we're a little worried when we first saw this graph because it's so variable, but as you can see, a lot of the lines are parallel. So when we have there's a big dip over here, so a cloud went by or something like that, and you can see that all of the lines go down there. So the next thing we looked at was, what does this look like when we process it with the index? But, do you have anything to say about this?
Yeah, notice that this is a sigmoid curve. So the closer you are to noon, the less affected you are by the changes of light during the day. So as you move farther and farther, this curve will start going down more deeply, and then you will need more frequent adjustments of the white control to adjust that. This is not adjusted by the white control because this is just looking up, so you cannot correct by a white control looking up.
Yeah, and yeah, around noonish here, it looks like there might have been some light cloud cover or something like that that was causing some variability. And I saw a question down in the box asking if this was measured in terms of irradiance. That is the setting we use on the spectrometer. But this actual data is not graphed off of the irradiance value. This is actually graphed off of the intensity value. So we're just looking at differences in the intensity. We didn't want it to do any processing, because when we set our reference using that irradiance measurement, basically it's going to set that as a maximum. It's going to give us kind of a percentage of that. But we can't control what the sun's doing when we start this. So if it gets brighter from when we take our initial reference, if we're using the radiance values, it's going to get saturated. So what we do is we set the integration time automatically, and then we knock it down from that so that we're at about 50% of what it says the maximum irradiance is. So if it gets brighter, it's okay, and if it gets a little bit darker, it's okay.
Okay, let me ask a little bit the question from Clay Sneller. The curve, if you have no clouds at all and not influence of anything at all, will be a smooth sigmoid curve. This peaks, this peak here is probably a cloud. This peak here is also some type of disturbance in the upper atmosphere, probably very high clouds that are passing through. And these wavelengths in the water area, in the infrared area, are very sensitive of anything that will absorb any water or humidity or wind that is coming up that is more humid. So those are the things that you don't control. But the very important thing that you need to notice here is that these lines are parallel. And this is very important because the water, we will be using normalized indexes. And that's something that we didn't thought that much. But if you think about the normalized index, so I'm pointing now to the normalized index that we use, the normalized water index 1. We use the 1 and the 3, that is basically different lengths. But it's always this, is 970 minus 900. So now, all these differences will disappear because the distance between here and here is almost the same as the distance between here and here or here and here. So, I'm sorry, I lost the arrow. So the distances will remain the same. So basically the use of a normalized index is already taking care of a lot of the variability of the downwelling energy. So that's why you will be seeing in the rest of our presentation and our analysis that we are not using much the downwelling energy. We are starting to learn how to use it, and it's not that simple. But even if you use the upcoming energy, you don't have a problem because the fluctuations in energy that are due to these variations in the atmosphere are already taken care by the use of a subtraction with the internal reference within the spectrum. And because the different wavelengths are the same, there's not such a big difference. You will see that Tyson will show you the trends once we do these corrections, but it's not as big as the trend that you see for the sun. So nothing. Keep that in mind. We are working with normalized indexes, so that is taking care a lot of our problems. And even Tyson was looking at the wavelengths in the 600, 700s for nitrogen, and these same dips that we are seeing in the 900s, you see them in the other wavelengths too.
Yeah, and to answer Clay's question really quick, auto setting the integration time basically sets it so that it's near saturation, so you get the highest sensitivity. So what we do is we automatically set the integration time so we know kind of what the maximum is, and then we turn the integration time down from that. This is for the downwelling. We turn the integration time down so that we can have the sun either get brighter or darker, and we can still detect that. Because if we just auto set it, it's going to think that's the brightest it'll ever get. So it's close to 100%. So we do need to turn the integration time back down a little bit.
If not, what happened, Clay? If you just use the very close to the border, is during the day there will be times that you will saturate the system. Let's say that you set your integration time at maximum here, where I'm putting the arrow. Now you move it during the day here, all these measurements will be saturated. The only way you will need to start changing again the integration time. And the problem is when you change the integration time, then the measurements change a lot. And we will show at the end of the presentation the effect of changing the integration time. But then you need to, your measurements will be affected by the change in the integration time a lot. So if possible, we prefer to keep the integration time fixed for the downwelling.
Okay. Looks like David's having a question. Once he's done with that, we'll go ahead and answer it. So here is a quick graph. At the times, they are, yes, they are daylight savings time. These are Pacific time. So this is what time is set on our watches and our cell phones when we're taking these. Down here is the analysis of our downwelling channels. So this is basically these same two lines we saw up here, this green line and this red line. I think, oh, yes, but these are processed with the index. So this is our two different indices, yes. So this green line up here is NWI1, and that's the 970 wavelength divided by, or minus the 900 wavelength divided by the 970 wavelength plus the 900 wavelength. So this corresponds to this blue line minus the green line over the blue line plus the green line. And the red one here is just 880 instead of 900. So it's 970 minus 880 over 970 plus 880. And as you can see, these are a lot less variable. There's still a little bit of bumpiness in here, but this huge dip that was over here is gone. It does a pretty good job of smoothing it out. But as you can see, there's still a little bit of a trend where it's kind of moving throughout the day. And if you notice, this looks more or less inverse to these. So while these are going up, these are going down. So that's something we need to kind of think about if we're going to be correcting our values with this.
So what we're thinking is that still the downwelling values still may be useful to do some precise adjustment. We have been playing with, and I have no idea from because I haven't read about it, or it's just a wild idea that we have. So tell us if you think that it's incorrect. Is we are trying to use the indexes from the downwelling, so the difference between the wavelengths, as a covariable in our analysis of variance, so we can take these trends and adjust them by there. But the problem is because the trends are different in different parts of the day, it is very difficult because an assumption of the analysis of covariance is that your regressions needs to be uniform across treatments. And here they go in opposite directions. So it's we're still not proposing that as a method to use. But if we can find a way to account for this small variation, then it's not a problem. If you are taking your whites frequently, then you will cut this variation and adjust in your upwelling measurements. You will be compensating for this variation in the energy during the day. So that's why you take the white measurements. And it's not that simple to make a correction. Let me go up here. Imagine now that I'm making a correction with this. So you will see that the upwelling values do not show this peak. So that when the plant reflects the light, it doesn't act so fast to a cloud or a thing that it's going through. So if you will say, well, I will just divide the light that is being reflected divided by the light that is coming in, or I will subtract, this peak here at 240 will produce a huge variation in your data. So actually, so far we have not been using the downwelling data to correct our reflectance data, because until we have a mathematical way to deal with these things, we don't know how to do it. So that's why we're saying maybe if we correct our data that is a difference, because our reflectance data is presented as an index, maybe we also need to correct using a difference of the downwelling thing. But it's not a simple function because it changes during the day. In one part of the day will be a negative correlation, in another part of the day will be a positive correlation. So it's not that simple. You will see when we start getting into the reflectance.
Okay. Were there any questions on that at all? Go on to the next one. So this is a little bit funny to look at. Basically, this is all one genotype throughout our blocks. And we have on the left side here, all these plants were in a watered block. And all the plants over here were in a dry block. There were 6 plants in each. And as I said, they were all the same genotype. And this is the raw intensity values that we were getting with the spectrometer. So up here, this top one that we're seeing is the 970 wavelength, and it's the intensity graphed over the time of day. So we're looking at kind of how it's changing over the day. This one here is the 900 wavelength for the upwelling. Sorry about that. So this top one is the downwelling. This is what's incoming from the sky. This one here is the 900 wavelength from the plants that we're getting. This is what's getting reflected. Here's the downwelling. So this is what's coming from the sky and the 900 wavelength. And way down here we have what's getting reflected of the 970 wavelength. One thing that's a little weird on here is that the 900 that's incoming is lower than the 900 that's being reflected. And this is because this isn't corrected with any reference or anything. So this is the raw values that we're getting. The reason that what's reflecting is brighter than what's coming in is because basically we have different integration times on each one. There's a lot more light coming into the top one, so we need to set the integration down a little bit so that it's not saturated. If we have them both at the same integration time so that they're seeing the same things, it just gets saturated and we can't tell anything. So it's not the absolute values that we're concerned with here, it's more the difference between the values. So that's kind of another reason it helps to put them in an index. And as you can see, through these two downwelling lines, the blue and the green, they more or less follow each other. They're still parallel, and to some extent, the 900, this orange one, and the 970, the black one, follow each other a little bit, although the trend's not quite as obvious. So we're looking into how that affects the index. Do you have anything to say on that?
Yeah, I will make a comment. So this dip here is the big dip that you saw. Let me go up a second. So this, we are looking at this big dip here. Okay. So this is the downwelling. The blue and the green are the downwelling, both of them showing the dip. But notice now the light reflected at the same wavelength. So this is 900. This is 900 reflected from the plant. So the plant is not showing at all this dip. The same thing is with the 970. So the 970, you have a big dip here. When you look at the reflected light, there's no dip. So if you attempt to correct your reflected light from your incoming line by a simple measurement of a division or a subtraction, you will create, if you divide by this number, you will create a peak here that goes up because you will be divided by a smaller number. So it's not that simple to use the downwelling reads to correct the upwelling values. Any questions for that?
Right. Yeah, Junli here said, how did you measure the up and downwelling simultaneously? Well, actually, when you push the button on the Jaz, it measures both of them at the same time. But the problem with that is it does both of them at one integration time. So for this, for example, I think we had our upwelling was set at an integration time of 32 milliseconds, but our downwelling was set at an integration time of around 9 milliseconds. So when we save an upwelling measurement, it's saving the upwelling and the downwelling channels at an integration time of 32 milliseconds. So our downwelling channel gets saturated. So what we have to do is we go through the plot, we take our 5 measurements, and then we start taking the downwelling measurements after that. So they're not taken at the exact same time, but they're all within a minute or two of each other.
We are trying to get Ocean Optics to answer, but they are not very responsive, to say the least. And but we try to see if they can solve that problem, and if they let us adjust the integration time of both channels independently, then it will be very easy, because you will click a button and you will get both measurements. For the time being, I will say that if you are short of money, maybe even if you don't have the downwelling, you will do relatively well, at least for the water indexes. We still don't know how to use the downwelling information. We want to have it because we wanted to play with it and try to learn. And there seems that there might be some trends that we can use to correct, but we still don't know how to do it.
Right. Were there any questions on that? Okay. So here is an analysis of the upwelling and the downwelling on the same graph. So these orange and green lines are the same as the green and red lines we saw here. But also graphed on this are the wavelengths that we're getting from the plants. So this
is taken when the plants, the spikes completely out, the grains is starting to form. So let me move you faster. This is the field. Can you see it, Clay? Right? So the very beginning of milk stage of the grain. So the leaves are still green. Okay. Let me tell you something, because if not, you will not understand. So this is an experiment where we have, it's not the typical association mapping experiment that you guys will be doing. This is just 2 varieties, Hahan and Attila. Hahan is more susceptible than Attila to drought. With the normal varieties have the complete 1RS translocation, and the other varieties, let's think it as a known 1RS, but it's actually a 1RS that is missing a couple of pieces that was engineered by Adam Lukashevsky. So we have genotypes, we have 6 blocks in each experiment, and we have 2 drought locations and 2 wet locations, irrigated locations within the same field. So there's variation between genotypes 1RS and non-1RS, and between varieties, and between irrigated and non-irrigated. So we take the water in the measurements in the morning. So when you look at the measurements here in this part of the day, these are our measurements on plots that have been watered. When you're looking here, these are measurements on the plots that have not been watered. So you can see there's a lot more difference among genotypes there. So that's what this variation here is the difference between genotypes. If you do this curve for individual genotypes, then it's a smooth line. So this variability is because this is an actual experiment, and this is the real signal of different genotypes responding differently to drought.
Right, and just on that same vein here, there's still a little bit of variability in the water, and we were actually seeing differences in our plants in the water too. They were watered a while ago, and some of the more sensitive ones were starting to show a little bit of a phenotype, even when we were just looking at them.
Let me ask to David Hall. No, a rotating device is not a good idea, because the most important thing that when you're taking your measurements is that let me go again back here. This part here needs to be perfectly perpendicular to the plot, because if you start pointing this one wrong, then you start taking measures from the soil. So you want this as fixed as possible, and actually we have here, we are not showing it, but we have a level on this part of this bar, so we are showing that this equipment is perfectly level and vertical, and this is pointing in a vertical direction. So if you start trying to move this around, it will be you set up yourself for a lot of trouble.
Yeah. yeah, we basically just have a post level on the side of that that vertical bar. So it's got two bubble levels on it, so we can make sure it's level in both the forward, backwards, and the left-right directions. So back to this graph. As I said, these two lines up here are basically the downwelling channels corrected with our index. So the orange one is basically NWI1, and the green one here is basically NWI3. And this blue line down here is our upwelling measurements for NWI1, not corrected with the white reference, and this purple one is NWI3, which is not corrected for the white reference. And down here, there are actually two lines. You can't really see them because they follow each other so closely, but this is our two indices, NWI1 and NWI3, after they're corrected with the white reference. And even though they're different indices, they follow each other almost exactly. I think there are a couple parts where they're a tiny bit different, but for the most part, they follow each other and they're uniform.
And the statistical analysis show exactly the same thing. So the index 1 and 3, they show exactly the same result every time.
Yep. Anything else on this graph? Yeah? Okay. So next thing we have here is just kind of an analysis of all of that data we were just looking at. So are we actually able to tell differences between these genotypes in the watered and the not watered fields? So on this graph, looks like my note over here got erased, but more negative is better on this graph. So more negative number means it has a better water status. And here we have our two genotypes. We have the full 1RS plants, which are more drought tolerant, and over here we have our 1RS recombinant plants that are missing whatever the piece is that is responsible for this drought tolerance. And these two lines are basically if these plants were in a watered area or a dry area. So as you can see in our drought-resistant plants, there's not really a difference between the watered and the dry treatments, and they have a pretty good water status. Over in the 1RS recombinant plants, the water status isn't as good as it was in the 1RS tolerant plants. So even in the watered plots, we're seeing a little bit of stress in the sensitive plants. But in the dry plots, we're seeing a lot of stress. The water status is very different than it was over in our tolerant plants, either in the dry or in the watered. I'm sure George had something to say about this.
So basically, we were able to detect differences between genotypes. We were able to detect significant interactions between the difference between genotypes and water and dry conditions in a factorial analysis of variance. So we can say that the differences between the two genotypes are mediated by the difference in watering. And actually, this is the average of the two different genotypes that we tested. The results are more spectacular in the more susceptible Hahan genotype than in the Attila genotype, but we can see significant differences in both of them, and in both of them we can see a significant interaction with the water and no water. So bottom line, yes, we can see the differences. You need to understand that this is a very, very clear difference. To be honest with you, in the dry environment, in the susceptible individual, we could tell it with the naked eyes that the plants were suffering. I measured the distance of the leaf that was drying up from the top, and I was able to detect significant difference even with that crude measurement. So it was a big difference. But the second genotype, we couldn't tell any difference by eye, and when we did the statistical analysis, we were able to tell the difference. But we have 6 blocks, and we have 2 experiments, so basically it's a 12 blocks experiment. So we have a lot of power. This is a very different situation to the one we are going to be facing when we do association mapping. The other advantage that we have is because we have blocks, we are taking basically, where is the arrow? the variation during the day of these small trends, because we are taking 4 measurements, new block, 4 measurements, new block, 4 measurements, new block, the variation during the day is also disappearing. It's been taken out by variation between blocks. So we are in an ideal situation, but in this ideal situation, it works pretty well. And this is not corrected at all by the downwelling information. This is just upwelling information using the normalized index and doing measurements of the white. How frequently, Tyson?
We're taking a new white measurement about every hour. We're doing it every 12 blocks. So we do 3 reps, and then we take a new measurement within the big block.
Any questions? It seems that it works, surprisingly.
Yeah, we're definitely able to tell differences, so that made us happy. Okay, so I know a lot of you are interested in nitrogen, so we took a quick look at some of the nitrogen indices, and one that I found was the 734 wavelength minus the 750 wavelength over the 734 plus the 750 wavelength. And I think this is actually a vegetative index, but it's highly correlated with the nitrogen status of the plants in some experiments that they did. If any of you want, I can send you that paper. I should have put the reference on here. Do that though. So basically, this is more or less what we were looking at earlier. You can see that throughout the day, these 2 lines track each other. The blue is the 734 and the red is the 750. And whenever one goes down, the other one goes down. Whenever one goes up, the other one goes up. We have the same big dip over here, even in this wavelength. So the cloud was kind of blocking light in all the wavelengths, it looks like. And then here we have the upwelling values of those 2. Once again, these are the raw upwelling values. These aren't corrected with the white reference. These aren't in terms of reflectance or anything like that. It's just the raw values that we're getting. And as you can see, we're still having variation between our genotypes, even though we're doing drought. We think this is because this is a vegetative index. So that might be something to think about when you are picking your indices. You might want to know if it's actually looking at nitrogen or if it's looking at the leaf area index or the canopy cover or something like that.
In our case, it could also be that the 1RS may have a larger root systems and is taking more nitrogen. We don't know. Yeah.
And there was something else we were going to say about this. Even in these upwelling, you can see that they kind of track each other. So over here, when this guy goes up and then this guy goes down, it kind of follows the same thing. It's a little harder to see the trends in here because there's difference between the different genotypes. Anything else on this? Yeah. Okay. So now here we're looking at the raw and corrected values that are processed with the index. So up here, this is our downwelling that's been processed with the index, and as you can see, it took out a lot of the variation. That big dip over here is gone. Kind of the squiggly stuff over here is gone. And then our blue line here is those same raw values that we had on that last slide, but these are processed with the index. So now what we're seeing here is kind of more the genotypic variation. And this red line is just the same values after they were corrected with the white
There's a question for when Juan Chao. I think that Tyson answered before, but basically the machine will take automatically both up and down measurements, but we cannot use it because it doesn't allow us to adjust integration time. So basically we take the 5 measurements from the light that is reflected, and then we take the 3 measurements from the light from the sky. So we take them separately. So they are not taken simultaneously. They are taken a few seconds after.
Right, and as George mentioned earlier, we are talking with Oceanoptics to see if there's a way that we can actually take them at pretty much the exact same time, because that would be ideal. So one other thing that we looked at, this is actually with these nitrogen indices. This isn't with our water. So when we were looking at this, we were a little worried because the two wavelengths were so close. It was 734 and 750. We were doing a boxcar of 10, and that's a pretty big boxcar when your indices aren't that far apart. So we want to make sure we weren't smoothing over the important peaks or anything like that. So what we did here is basically all of these top guys and all these bottom guys are the same thing, except the top ones, when we took the 734 wavelength and the 750 wavelength, we took an average of that wavelength plus 10 wavelengths on either side. And that's these top ones. So that's a boxcar of 10. And then on the bottom, we just took the closest wavelength to 750 and the closest wavelength to 735. And that's what's on the bottom. And what this is each genotype in each kind of block. So here we have our Attila genotype. The red line is the Attila variety. The red line is the 1RS recombinant variety, so that's our drought-sensitive one, and the blue line is the full 1RS variety. That's our more tolerant one. So this kind of reflects what we've been seeing with all of our data. These two graphs here are Attila, and these two graphs here are Attila. And we're not really seeing much of a difference in those for drought, and even here in the nitrogen index, there's not that big of a difference between the two. I mean, especially over here in the water, you can't really tell those apart. They're kind of all over the place. But for our Hahan, this is the same thing. We have our recombinant one, which is sensitive up here, and our full 1RS, which is tolerant down here. You can see that there is a little bit of difference here, and the difference is bigger in these dry ones than it is in the watered ones. And if you look at the difference between each graph for each variety and genotype for the treatment, there's not that big of a difference between using a boxcar of 10 and using no extra boxcar. So these graphs look pretty much like these graphs. These ones look pretty much like these ones, and so on and so forth. The only really big difference that I noticed when I was looking at these, and it's not even that big of a difference, is second guy over here. He jumps up a little bit more when there's no boxcar. But all in all, it didn't look like there was that big of an effect using a boxcar of 10 or a boxcar of 1.
I think I will clarify one aspect. We are talking about two different boxcars. One boxcar that is taken automatically by the equipment, and that's the boxcar that he mentioned in this slide. This is this boxcar. This one is automatically, once you set that number, the equipment will average that wavelength with as many as on the size as you mentioned, and take that number, and that number will be the number that is here. The boxcar that Tyson was talking about is then when we process the indexes, and you need to say, well, let's say I'm going to set an index that use 494. But if you look here, I have 3 measurements for 494. So which one I use? So you can say, well, I will use the average of these 3. So I have the 494 nanometers covered. Or you can say, actually, to be sure that I don't have too much variability, I will take for the 494 from 492 to 497. So that's the boxcar that Tyson was referring. In our water indexes, when we did the calculations for the 900, 97 minus 970, that 900 is 900 plus minus 10, correct, Tyson? So you have...
Yeah, so plus or minus means, 10 on each side. It's not 10 nanometers.
10 measurements. So that means it's 3 nanometers on each side. So when we said that we are measuring 900, basically we're doing an integration of the wavelength between 888 and 902. So by making the average of those two, we are kind of taking the not just the peak itself, because there may be some variation, but we are taking a little bit of the area on that region and making an average. So that's the manual boxcar that we use when we decide by hand how we are going to calculate the index, the water index. Questions?
Okay. Okay, so yeah, that's about it with the boxcar. We didn't see huge differences in our nitrogen index that we were looking at. I haven't done this with our water indices yet, but we'll probably see about the same thing, maybe less of a difference. If you guys want, I can let you know what our results are on that. Next slide we have here is what our right references look like over time. So when you take a measurement, you get all those columns, and there's one, the R column is the reading you get from the reference, and that's in terms of the absolute intensity, and that's what you use to correct in terms to put your values in terms of percent reflectance. So this is basically how they were changing over the day. As you can see, there's a lot more variability up here in the lower wavelengths, up to about 800 nanometers throughout the day than there is around 900. So this area is where we're doing most of our water indices. The two different ones we have are 900 over 970 and 880 and 970. So there's not a whole lot of difference here throughout the day, but there is definitely some difference, especially in these earlier wavelengths. And it's a little small here, but you can all download this PowerPoint presentation afterwards. And basically each line is a reference taken at a different point. And yeah, I took one at 10:50 in the morning, 12:20, 12:30, 2:30, and 3 o'clock-ish. And yeah, a lot of them follow each other. I think there's one, the 10 o'clock one is lower than all the other ones, but more or less they follow each other, especially in these higher waves.
It's nice to see, because you see what is your Y control. This is you are taking percentages of this, so also you see why your percentages will be a little bit higher in the 900, because you are dividing by a smaller number also.
Right. Yeah, so when we were first looking at our graphs, we were wondering why our 900 value was higher than our 975 value, but that's because there's more light at the 900 wavelength. Okay, so this is just a little bit of a reiteration of those first couple graphs we saw. This is raw values for our downwelling, but this is from a separate experiment from that first one. So that's why you're not seeing this huge cloud drop over here that we saw. But what we are seeing on this one is this huge jump up here. And what happened here was we changed the integration time when we went from this reading to this reading. So when you change the integration time, it does definitely affect what's going on with all your values. Is one reason we think we should try and keep the integration time the same. Even so, you see all of the lines track each other as they do with all the other variation in here. And these are just the same water index values that we were looking at earlier. Did you have anything to say on this?
These are the raw values. So when you go later to the, you have the subtraction of the indexes. Yeah. Okay. Yeah.
So this is what they look like after they're corrected. So those three lines that we looked at just now basically collapse down into these two lines when we put them in an index. So it takes away a lot of the variation. You can't really tell where we changed the integration time, but you still feel it would be better to keep uniform integration time, just to help eliminate any variation.
This is a different day. Again, we are measuring the water first and the dry part second. That's correct, Tyson? But it's a different part of the field than the one that you saw before, but you see the same trend. Little variation between genotypes when there's water, and a huge variability between genotypes when there's no water. So the genotypes are sensitive to drought. And you can see that the downwelling here, this experiment was all done in the afternoon. So from 2 in the afternoon to 4 in the afternoon. And you can see that, yes, there is a small trend here in the energy that is coming, but you kind of don't see that much in the upwelling. So I don't know, still I don't know how much we are going to use this to correct.
Right. And then I think we have one other graph here. This one's a little tough to look at. This is basically just those 3 downwelling measurements are these guys that are tracking each other, and you can see the jump in the integration time. And these other ones are the same wavelengths, but in the upwelling channel. And you can see the it's basically the same thing we saw before. They're following each other. They jump up when the other one jumps up because of that change in integration time. I don't believe that the integration time was changed as much for the upwelling as it was for the downwelling in this. So that's why you don't see quite as much of a jump in these guys as you do.
Here it's difficult to see because the variation on the integration time is hidden on the variation between the genotypes.
Right. Yeah, so it's a little tough to see it here. But once we apply the indices, you can see it's more or less removed. So Mike has a question. When you say dry, what sort of yield level are you estimating?
Yes, I actually do know. We did the experiment in 2009, and we have a 1 month of drought, so without no rain between March 1st and March 31st, and then we have a rain in April. So what happened is the 1RS line that never was stressed, and the other line has visible symptoms of stress similar to this year. The difference were in the Hahan, 800 pounds per acre difference in yield. So in the Attila, no, it was 1,000 in the Hahan and 800 pounds per acre in the Attila. So huge differences in yield between the two. So in the drought, you are starting really a significant impact. And in the other ones was a normal yield that we will have, I don't know, 5,000, 6,000 pounds per acre for experimental plots. That answer your question, Mike? So Mary has a question. If you need a microphone, sometimes just ask Diana, and she can open the microphone for you if you have a complicated question or you want to make a point. We don't want you to be all day typing. Yeah,
I think we're pretty much done with the presentation, so let's just take questions now. So Mary has 2 practical questions. One, is it really tough to see this screen in the sunlight? Any hints? And second, can you make us a new tutorial to walk through the menus? Yeah, we can make you just kind of a quick tutorial to go through the actual Jaz menus. It's not very complicated. It should be pretty easy. As far as your first question, I'm not quite sure what
you
mean.
If you consider if the reflection of the light it's not letting you see the graph in actual real time.
Oh, the screen on the spectrometer. Yeah, actually with that Jaz pack, it has a nice little built-in flap on it so that you can actually, the screen is actually shaded, so you don't have to kind of use your hand and try to look at it. We didn't have too much trouble.
It's an expensive pack, $75, but it's what's worth it. It has the special holes to put the connectors, and it has the flap to cover. So I think that if you are going to order this equipment, it's better to spend those $75 and order the band. That was something that was useful.
Yeah, and it has the strap on it just so you don't accidentally drop it or something, since you don't want to be dangling by the fiber optic cords or anything. And yes, you can close the lid on the bag. It just has this little kind of flap that flips up. It's still connected, and it kind of shades it. And then when you're done, you can put it down, and it kind of velcros back on.
Everybody's typing, so let's see who gets it as first. This is kind of fun. Mike, can you send specs for ordering a new instrument? Well, we did send the instrument that we got, and I sent to Luther, has received all the things from us again. I can send the order that we made, but yeah, it is, yeah. We will put it on the we'll put it on the. I'll send it to you, Tyson, and then you put it on the discussion group or whatever in the website. Huh?
Okay.
So the
yeah,
things are the important things are the gratings. So in the drought tolerance, we are using grating number 4. And I think Clay is ordering number 3 to be able to measure on the lower wavelength. Then you need to have the cables. We found, at least for California, we have to change the cables, the fiber optics, because they were too wide. So now we're using, what fibers are you using now, Tyson? Up and down?
We are using a 200 micrometer fiber for the upwelling and a 100 micrometer fiber for the downwelling.
The white control that they sell you for $300 is absolutely useless because it's 3 centimeters squares, so you cannot use it at all, so don't spend the money on that. The Jaz programming language is garbage, so don't spend your money on that. You cannot do absolutely anything with it. And what else?
Yeah, we haven't used the Spectra Suite software either. We did all of our analysis in Excel, and we didn't have any problem with that.
So Junli is asking about the post-processing of the data. So hopefully, once we have a programmers in place, we can write a script. I have a student that is very good that can write a script, but he's extremely busy finishing his PhD and leaving the lab. So I'm not sure if he will have the time. But it's not that difficult, because the file is just a text delimited file. So we needed somebody to write a script that will parse those files, get you the averages, and transform everything into an Excel file. So then it will be very easy. But we are not at that point yet. We will have the programming power once the people is higher in Cornell, but we don't have it right now. So Tyson has been doing manually, just by you can open each file in Excel and then take the column that you need. So basically, you can take the process column. Let me show you something. The first column is constant. So what you can do is, for an experiment in the field, you can open an Excel file that will have the first column will be the wavelength, and then for each plot, if you don't want to keep a record of the dark and the white, you can just have one column that is your process value for that particular plot. So at the end of the day, you will have one column for the wavelength, and then each column of your Excel file will be one plot. So then it's very easy. If you want the 900 lengths, you just go to your Excel spreadsheet and you get that number. Once we decided that we will make the processing automatic, in our mind, in that program, it will also give you the indexes that you want at the bottom. But we are not at that point right now. Let me try to see.
Yeah. One other quick thing about that is for that first column, for a given spectrometer channel, it's always fixed, but the numbers are slightly different between our two different spectrometer channels because of the way they're calibrated. So if you do have two different channels, you'll just have to keep that in mind when you're processing the data. But for a given channel, it's always going to be the same way.
Kleisner is asking again the question, how do you get it read for both channels? It's automatic, but it doesn't work. So right now, the way it's programmed, you press the button and you get the two reads. And it creates two files automatically. But the file that is upwelling, it doesn't work because it's used the integration time of the downwelling. So right now what we do is we take separate measurements. We take 5 measurements for the reflectance, stop, change to the other channel, and take 3 measurements from the sky. They are separate ones. Then Colorado State. For our trailer run this summer, we are thinking of taking leaf samples to measure leaf water content at the same time we take the Jaz. Have you done that? No, we haven't taken measurements for the leaves. We plan to take from this experiment a grain and do a carbon isotope discrimination to see how it correlates. David Hall, are you using the same gratings for upwelling and downwelling? Yes, we are using the same grating for both of them. Mike Pumphrey, can you do some calculations to determine what level of power we need, assuming we don't have the same level of replication as your experiment? A way in which I'm maintain significant in your trial. Well, for the hand difference between a very susceptible and a very resistant genotype, with 6 reps we saw it without any problem. Probably we'll see it with less. The problem is when you do an association mapping trial, you will only have replications for the checks, but you will have no replication for the rest. So I do not know, Mike. I do not know. And so here we are in an ideal situation, two genotypes that are very contrasting between each other, so contrasting that we can, for one of them, even see it visually. And we are detecting huge in the hand. The difference are p 0.00001. So probably even with 3 reps we would have detected there. But I don't know. I don't know how the association mapping will work. I have my own doubts on that. Kleisner, my question seems likely it will vary by the index you use. No, I think it's not depending that much on the index. Well, the index will affect, but the index the variability was not there. The variability is between plots and plants and in the field, and one plot will have more access to water, or there will be something between the soil that the roots will be able to get to more water than the other, or will be closer to the irrigated plots that are close to it. We did an experiment in Imperial Valley, and we saw an effect that we imposed a drought treatment, but it was close to another field that was irrigated. So up to 2-3 meters from the site of the irrigated field, we were seeing that the plants, as you're getting closer to the irrigated field, were getting better. So there was some water that was coming from the irrigated field into our drought experiment. So there are a lot of factors that create variability that are more than the index. The indexes are pretty stable, actually. NAY1 and NAY3 are exactly the same. And actually, I ran all the other indexes that Tyson can show me, and I found significant differences between genotypes for all of them. George, Jada Star Martins. George, can you please put your thoughts on the Jaz script language and what you need it to do? Well, we send a lot of questions, but we never receive an answer, Jada. So we will need to program the integration time. We will need some control of the files. Tyson, you want to tell Jada something?
Yeah, one of the biggest problems that we're having with the programming language is that it's very hard to handle the file names. we could program all of our different integration time and take the upwelling and the downwelling at the same time, and figure out how to do all that. But when it comes to actually saving those measurements, we can, there's a function in there so that we can increment the file name for that one session. But as soon as you turn the unit off and then you turn it back on, it's just going to start overwriting all of those files. So if either you have to bring a bunch of SD cards out there with you and pop a new one in every time you turn it off, and then it's going to be a nightmare trying to figure out what's what later. And there's also some things, from what I saw, there wasn't any way to put timestamps on the files, or basically get anything like what you get with the normal output. You're losing a lot of that extra information if you're using the programming language, it looks like.
But you will be pleased to put those things in writing and send it to you, so you can transfer to your engineers. I think that the most urgent thing that will help us a lot is if we can change the integration time for the upwelling and downwelling by ourselves, and then when we click it takes the two measurements. That would be fantastic for us. Just that simple modification would be fantastic for us.
Yeah, I don't think we'll have too much trouble doing that. It's just the actual saving of those files that they're having trouble with right now.
Jaden, do you want to have the microphone to say something?
Okay, let me see if I can do this. Actually, Jada, what I'll do... Oh, there we go. Enable audio. Okay, so you should have a microphone now. So you can just speak and we should be able to hear you, Jada. And to be able to talk, you have to turn on the microphone icon at the top, so it's on green. Okay, George. Let me get you guys back on.
Hello? Can anyone hear me now? No? Okay, so you need to change, do you need to change the integration time for each channel independently? I can't hear any of you now.
Sorry, I had to connect it again. Yeah, she gave us authorization by me to connect it again. I thought it was going to be automatic. So everybody has mic now. So now you guys can complain if you want. Nobody wants to complain. Jada, can you hear me? You can talk now. I think that everybody has a mic now.
Well, Jada, basically what we want to do is we want to take a downwelling measurement at one integration time and an upwelling measurement at a separate integration time, one right after the other. And then we want to save both of those.
Oh. Okay. Anybody else have any questions that you would like to see? It takes some time to learn all this, and you have to play a little bit with it, and it will be a little bit cumbersome at the beginning. The post-processing is cumbersome, so we hope that we can help you guys a lot. The measurements are relatively fast, so Tyson was able to do 48 measurements, or let's say the 24 for one experiment in how long? A couple of hours.
The first time it took a little longer just because we're getting used to it. The second time, I think it took us about an hour for 24 measurements, or well, 24 plots. So we're doing 5 measurements for each one, and I was also doing the downwelling. So it'll be a lot faster if we can get the programming working.
What about the possibility of using different fiber sizes so that you could get by with the same integration time?
We thought about that one, but it will change with days. The problem is you will find that we probably can do that, and you don't have that many options of fibers. But then it may work one day and then will not work the other day. So we need a more permanent, more flexible solution.
It looks like Judy had a question. Is it possible to measure just one wavelength for water and one wavelength for nitrogen and get adequate results for each phase? When you take a measurement, it saves. Okay. When you take a measurement, it actually saves all the wavelengths. So you have all that extra data, and basically we're going to try to write a program that'll process all of that data. And if you want, hopefully we can make it so that you can just put in the wavelengths that you want, and it'll extract those for you, and then you can process. You use whatever indices you want.
The idea is to store in the database the 2,000 measurements, so that anybody can go later and analyze the spectrometer data in any way they want. And that's the beauty of this equipment. That's why we went here. You can ask a lot of new questions, and as we learn more about what information is in each wavelength, we can probably extract a lot more information from our experiments. That answer your question, Judy? No, Judy, you need to have more than one wavelength because you are using normalized indexes. So you are using the differences between the wavelength. Basically, it's like you're taking an internal control in one part of the spectrum that is not of your interest, that is not water, and then your measurement in the 900 or 900 that is water, and then you refer one to the other to get rid of all the other variations. So you can. then it will not be a normalized index, and you will be subject to all the variation that you saw in the raw data. So the normalized index helps you to eliminate the result. Colorado's third. Are 5 measurements per plot too many? That seems pretty slow. Yes, they're probably too many. We haven't done. I don't have the time right now, and we don't have the power to do the analysis. But we can run an analysis of optimum subsampling. We have the data of the subsamples. We just run the analysis with the average. But we can run an optimum allocation time. But it's difficult to determine the cost of each subsample measurement. We did 5 because we wanted to be as precise as possible. Probably we did a couple of measurements, so 2-3 measurements per plot, which is more than sufficient. Okay.
Clay has a question. What about wind and a moving canopy? Is that an issue? I don't think it is. One of the days, the second day we went out there, it was very windy. The canopy was blown all over the place. But we still got
in this regards, this seems different than canopy temperature, because canopy temperature measurements, you are dead with a little bit of wind. And this seems a little bit more resilient to variations in wind. It seems to be correlating well with the water status of the plant rather than the temperature. We didn't did measurements of canopy temperature determination, but I did for this same experiment the previous year, and we saw a little bit of a difference, but there was more noise, not at all the level of significance that we are seeing in this experiment. So it seems that the canopy spectral reflectance is a little bit more powerful than the canopy temperature. We are going to see what happened, but we are going to do carbon isotope discrimination, and then we'll have those data to share. We were sidetracked by other things. We measure in all these plots roots. So we went 6 feet under the soil and take root samples of all 24 plots. So it was not an easy undertaking, and we didn't find anything. So we spent a lot of time washing. Well, Tyson spent a long time washing roots, getting things. We get my special machine, so we were not taking many measurements besides that, because we have a huge hopes on the roots differences, but we couldn't see them in the field.
Yeah, and we were planning to go out there and take some more measurements, but then it started raining like crazy here, so the field was muddy, and then yeah, a whole bunch of things happened. Mike asked if we're doing carbon isotope discrimination on grain or vegetative. I believe we're doing it on grain.
We don't have the resources to do that.
Okay. Are there any more questions?
I think that what we are going to do is then we will put the information there. I will need to ask you something. It's not nice, but I will try to direct the questions to Tyson. I'm leaving for a month to several meetings for the TCAP in Europe, and I'm at the very limit of what I can do. I have to finish the 2 grant proposals and other things in 2 days. So please protect me a little bit, because I'm taking hits from many different parts of the project, and I really need to receive less than 200 emails per day to be able to survive.
Okay. Thanks for helping out, Josh.
Okay, so send all the questions. Tyson will try to answer. If he has an answer, he will come to my office. I will discuss with him, and I'll give you our best answer. But as I said, we are not specialists. This is our first shot. We are still pleased that we were able to see the differences. So at least it seems that the equipment is working. Sometimes the equipment gets frozen, and then so we have to restart it. Tyson, you want to make a comment for Jada about that? How frequent?
We did have a couple problems. I think the first day we were out there, it did freeze, and Josh Hagerty, he was down in Imperial using this, and he said it froze, and he had to restart it a couple times. I did notice the one time it froze, it looked like the SD card may have popped out, so that might be what the problem is. We'll just have to watch out for that. I'll let you know if we ever run into any more trouble with that, but so far it's been okay. So if there are no more questions,
okay, nice to see you, all of you, at least the names of everybody here. It seems that this is a useful type of session to share information. So if at some time any of you have something that you think is useful, just call a conference, and those that are interested can come and learn from the others. So I think that will make us a lot more effective in the way we do our things.
Yeah, this is Dina. We have recorded this, so we'll be sending out the URL so you can watch it later or send it to other people who couldn't attend. And if you are interested in doing a session like this, you can send me an email and we can do a practice run beforehand so you can see how all the buttons work and things.
So it was very good, Dina. There was a large number of people, so it worked very well.
Yeah.
Jada, thanks for coming to the meeting. Let me know if you have any additional questions or needs. I will try to get them addressed. Thank you very much, Jada.
Yeah, I'll send you an email after we're done with this.